So now what we're going to focus in on is directional selection and natural selection affecting allele frequencies. And we've already talked a little bit about these. These are the ones in New Mexico, right? And we've talked about those genes that influence melanism and matching coloration, and we know the genetic basis of that. And we probably feel pretty strongly that natural selection can explain this, right? That you can see this background matching where you find good background matching in the middle of A and poor background matching in the melanic ones. And we imagine natural selection favors that particular phenotype, the genes that are associated with it, against different kinds of background matching, and it's presumably due to selection. So we feel pretty strongly that we can understand that, and natural selection probably explains that matching where, this one doesn't match so well, right? This one matches a little better. This one probably matches the best, okay? So can we imagine if there's a genetic basis to that variation, that trait, natural selection has influenced the frequency of those melanistic genes that influence coloration. So how do we talk about this? So what we're going to do, we're going to introduce what we call fitnesses for what we call a dominant locus. And we'll be talking about how we determine fitness in the example I'm going to show you a little bit later. So what we're going to say here is there are three genotypes again, A1A1, A1A2, and A2A2. This is the case. There are lots of different ways you can determine this, and we'll be talking about how we determine this. But what we're saying is in this particular case, A1 is dominant to A2, not necessarily in terms of coloration, but in terms of what we call fitness. And do you guys remember what fitness is? And do you remember what does that mean? Yeah, it's the reproductive success in general. It's the contribution of offspring to future generations of each genotype relative to the other, right? So that's the best way to do it. A lot of times what we do is we look at survivorship of these different genotypes or phenotypes, and kind of the mouse example. So there are different ways we can measure fitness, and we'll be talking about a lot of them, but always remember that fitness is always a relative term, okay? And in this particular case, what we're saying is this homozygote and this heterozygote have equivalent fitness. And so that in some sense, A1 is dominant to A2, okay? So this is the case of, this is what we call dominant selection. What about the A2A2? It has a reduced fitness relative to the other two. And this thing, we'll be doing this a little bit. We'll be showing you how to calculate this. This is what's called the selection coefficient. So if the selection coefficient were 0.8, what would the fitness of that genotype be? 1 minus 0.8 would be 0.2. So that would be a case where that individual, that genotype, had a fitness that was 80% reduced relative to the other two. So that would be what we'd call really strong. That's a high value. That selection coefficient's really high. What if that S were 0.01? Then the fitness would be 0.99 and not a big difference. So that selection, that S value basically tells you how strong selection is acting against genotypes. And again, we can calculate the change in gene frequency in one generation is given by the following equation, and it's derived. And I'm not going to ask you to memorize that, okay? Because that's fruitless, and you'll just forget it, right? What we're going to be showing you is the outcomes of this model, and then I'm going to be showing you kind of a cumbersome way to do it through a work problem, and it gives you the same result, but it's a little easier, I think. But this is a simple equation for any kind of selection model, for the most part, that you can calculate how much an allele frequency changes over time. If I were to ask you, what do you think the outcome's going to be here in this population with these kinds of fitnesses? What do you think is going to happen over many generations? What do you think the final outcome will be, assuming just selection is occurring? Huh? Yeah, A2 will be eliminated, right? And A1 will go to 100%, will go to fixation. Right? If this is how it works, right? That's going to be the outcome. Now the question is, how quickly does that occur? So let's. This gives you an example of this, and a lot of what we're going to be doing, you'll be doing this in lab in week 4. I can't remember which lab. You'll be getting introduced to a little bit with the bead example, but you'll be doing what are a lot of simulations. So let's go, if we go back to that other model and use the exact same thing, what we usually track is we start at some starting point in a simulation model, generation 0, and this is over 400 generations. Along here is P, which is the frequency, in this case, of the A1 allele, right? So we start these simulations where that favored allele is rare in a population. So maybe this is the case where this new melanin gene mutation arose 3,000 years ago. It was rare in a population, and those individuals got into these, you know, dark lava beds, right? And so what we might expect, what we can see here is what happens to the allele frequencies under these different kinds of selection models that I just showed you. What's the outcome in general, for the most part, after about 400 generations? What's happened to the frequency of a favored gene? It's gone up quite a bit, hasn't it? Right? In some cases, it's gone to fixation. Other cases, it hasn't quite gotten to 100%. So this is where it's rare. This would be the point where the A1 allele had gone to fixation, or 100%. What's different about the three different curves? Yeah, this one, for just a slice at any time, let's start, this is a 50th generation, right? This one is slow in terms of the rate at which that favored allele is increasing in the population. It's a much slower rate. Which one has the fastest? The orange one. And this is the case of the S equals 0.7. So in which case is selection favoring the dominant allele much more so? Which example is favoring where the dominant allele selection is favoring it the most, or there's stronger selection acting against the recessive? Yeah. This one, right? This is the case. S equals 0.7. That means the relative fitness of the A2 genotype is what? 0.3 compared to 1 for the other two. So in this case, that favored allele increases really rapidly early on, right? And it goes to fixation pretty much by that point, within 150 generations. It goes to 100%. And what's happened to the A2 allele? It's eliminated, okay? This one takes a lot longer because selection isn't as strong, favoring it or favoring the A1 allele, or selection acting against the A2 allele. So it'll eventually get there, and it takes a while. We're going to explain, what's the other thing you notice about these curves that all of these kind of share in common? This one is slower, this one is the fastest, this is stronger selection, this is weaker selection. What do you also notice about those curves? Yeah, they plateau or they asymptote, right? So what you can see is there's really rapid increase in the favored of allele, and then it really slows down a lot, doesn't it? We'll come to that, okay? So this is kind of what when we think about selection, what we're looking at is how do frequencies of favored alleles change over time, how quickly does it happen, and how long does it take to get to fixation? And we'll be talking a lot about why this asymptoting or plateauing occurs under this kind of selection. So we'll be coming back to that. So basic take-home message, the larger the selection coefficient, the stronger the action of natural selection. There you go, what I just said. Okay. You know what I think I'm going to do? Yeah, here's what I'm going to do. I'm going to mix it up a little bit. I'm going to talk about this slide, and then the beginning of next week, I'm going to go through, or Thursday, I'm going to go through the example of how you calculate allele frequency change in one generation by the manual method that I do. Okay, and that PDF is up on Moodle. So kind of follow, so look at that thing. So look at that let's just see how it's done. I'm going to do a similar problem with similar kinds of numbers, but where I don't have what's called dominant selection. So we're going to kind of compare those two. So let me introduce this idea first. I'm not going to get to A1 recessive. I'm going to go through these two on the left. And then I'll work through a problem on the iPad calculating the change in allele frequency after one generation of selection. So look at that PDF on Moodle. I'm going to do a slightly different version of that, but the exact same steps. And that'll be your second homework problem, or third homework problem. I think I'll be rushing to try to get through this in 10 minutes, and that's not good. Okay. Is there a video on there? Oh, good. Okay, yeah, watch that. Okay, so watch that. Okay, okay. Okay, so yeah, watch that video so you know how the calculations are done. Then I'll do a slightly different one, and we're going to compare what I do in class with what that video, the answer it gave you, and it'll illustrate these things. Okay? So let's talk about, this is the example we just showed. This is A1 dominant selection, right? The A1 genotype and the heterozygote have the exact same fitness. And we'll be showing you how to calculate fitness based on survivorship and all those sorts of things, so what these things mean. And then the A2 has a slightly reduced fitness. So what's the value of S in this particular example? Yeah. Fitness is 0.8, so 1 minus 0.8 is 0.2. So this would be a case of a selection coefficient of 0.2. Okay? What's different about this one? It's kind of like partial dominance, right? Remember when you had the pink color, this is in terms of fitness, right? This is the case of incomplete dominance, where the heterozygote doesn't have the exact same fitness as this homozygote, right? It's not up here at 1, but it's higher than this one. So this is what we often call incomplete, co-dominance, partial dominance. We use all kinds of terms, and it's not consistent. But this is the case of what we're going to say incomplete dominance, where the fitness of the heterozygote is slightly reduced compared to that homozygote, but is higher than the alternate homozygote. Okay, it's intermediate. Okay. I'm not going through this one yet. Let's go through this one. So here's the red line for this one. Here's the blue line for that one. What's different about those two? What's similar about those two curves? Again, in both cases, the A1 allele is the favored allele, right? And we started out at low frequency, so this is a favored allele starting out at low frequency in the population. And what we both see is a really rapid increase, don't we? Okay? So it goes up really fast because selection's favoring it. Remember, the scale is a little different here. It's 0 to 1,000. That's how all your simulations in the lab will be done on a 1,000 generation scale, so you have to keep that in mind. Okay? The other thing that's similar, though, is they both sort of plateau again, don't they? It takes a while. What's different between the red and the blue line, though? Which one goes to fixation faster? Yeah. What you can see is this thing doesn't have a very long asymptotic phase, does it? Or plateauing phase. Whereas this red one takes a while. It goes to fixation about right in here, 400 generations. So this model, where there's incomplete dominance, A1 reaches fixation most rapidly when it is incompletely dominant with A2. When A1 is dominant, this case, its initial increase in frequency is the most rapid. You see that? But its pace slows down once it is common in the population. Why? What's happening here? What's happening is, it's hard, what's happening in this, the A2 allele is eliminated much more efficiently in this case, right? It's harder in this case. See here? Why do you think it's harder? So here, now you're getting up in here, right? In this allele, the A1 allele is really common in the population, right? Why is it hard to get rid of the A2 in that case, whereas it's really easy here? Yeah. Yeah, so the key here, and we'll come back to this, and I'll be showing you an example of this when I do my work problem. What happens here is, as this A1 favored allele gets common, right, it's really high frequency, it's 98% in the population. Guess where A2 in a genotype is mostly found in that case? Guess what other allele it's associated with, or what genotype do you think it's mostly in? The heterozygote. This is what we call masking. So in these, it's really hard to get rid of really nasty genes under complete dominance, right? Because that A2 allele, selection can't see it, right? In this population, when that A1 becomes really common, A2 is usually associated with A1, and it doesn't have a fitness difference. So it turns out it takes a really long time to remove a nasty, deleterious gene when it's rare under this kind of model. But what about this one? See, it's got a reduced fitness. Selection, I hate to say this, I shouldn't use it, but selection can kind of see it, right? There, it has a reduced fitness, so selection can kind of get rid of it. It has a lower fitness. Okay, so what I'll do next time, I think that's, does that make, these are all kind of conceptual things. It takes a lot of practice thinking about it, and we'll be going through a lot of examples of these. But what I'll be doing is, I think the video that I gave you is the case of, I can't remember. It's either complete dominance, and I'll do the opposite one of incomplete dominance. I did codominance first, so I'm going to do the one of complete dominance then. Okay, I got it figured out.
You might have to check that.
I'll check it. Okay.